Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Have you ever stopped to wonder how a single, unspecialized cell, say a stem cell, receives its marching orders? To become something as specific as a neuron in your brain, or a muscle cell, or even a bone cell.
It's one of life's fundamental mysteries, isn't it? And it's crucial. When that biological programming goes off track, it can lead to some really serious diseases.
Absolutely. It's just astonishing when you think about it, that innate intelligence inside every cell.
Exactly. Navigating this vast instruction manual to find out exactly what they're supposed to be, scientists have been trying to decode that for decades.
And today, we're actually diving into some brand new research that shines quite an incredible light on this very process. We've got a fascinating health news piece and a detailed scientific paper. Both just came out this month, August 2025, from Case Western Reserve University. So our mission today is to kind of pull apart this complex science. We want to understand a really surprising new discovery about how our genes get regulated, and ultimately reveal what it could mean for treating some tough conditions, like certain neurodevelopmental diseases and specific types of leukemia.
Okay, so maybe think of your DNA like this enormous instruction manual for your whole body, right? Every single cell has a copy.
Okay.
Now, epigenetics, that's like this really sophisticated system of, let's say, sticky notes or maybe highlight markers on that manual.
Sticky notes. I like that analogy.
Yeah, they're not changes to the DNA sequence itself, the actual letters, but they're chemical modifications around the DNA. They tell your cells which pages to read, which ones to skip, essentially turning genes on or off, or sometimes just adjusting the volume,
Fine-tuning it.
Exactly. And what this new research reveals is a truly unexpected relationship, a synergistic one between two of these really critical sticky notes. It's not just about one marker doing its job. It's about how the combined absence of two specific marks completely flips the script.
That's the real twist here, isn't it? The combined effect. So let's unpack this surprising discovery. We need to talk about the key players, this epigenetic duo, as the researchers call them.
Right.
The study focuses on two specific epigenetic marks, H3K79 methylation and H3K36 trimethylation. Now, those are definitely mouthfuls.
They are.
Maybe for our chat, we can just call them the K79 mark and the K36 mark.
Sounds good. Simpler is better. So these are tiny chemical tags, and they get added to proteins called histones.
And histones themselves are pretty cool. They're like the spools that our DNA thread wraps around, right? To keep it all organized and packed into the nucleus.
Precisely. It's an incredible packaging system. And these K79 and K36 marks, they act like crucial volume knobs placed right on those histone spools. They basically dictate how loudly or softly a particular gene nearby gets expressed.
So they're telling the cell, read this section carefully, or maybe just skim this part.
Exactly that. And these volume knobs, they don't just appear randomly. They're placed there very precisely by specific enzymes.
Ah, the molecular machinery doing the work.
Yep. For the K79 mark, the enzyme is called DOT1L. And for the K36 mark, it's an enzyme called SETD2. Think of them as the cellular architects, carefully placing these tags to fine-tune everything.
Okay, so we have the marks, K79 and K36, and the enzymes that put them there, DOT1L and SETD2. Now, where does the surprise come in?
Well, here's where it gets really interesting, almost counterintuitive. The standard thinking, the dogma really, was that these marks individually generally help turn genes on. They promote gene activity.
Okay, that makes sense. Mark means go.
Right. So the logical assumption would be if you removed one of these marks, the gene associated with it should turn off, or at least get quieter, wouldn't you think?
Yeah, you'd expect things to shut down. Remove the on signal, you get off.
Exactly what you'd predict.
Yeah.
But the researchers at Case Western Reserve, including graduate student Cheng Zhang, who really drove this part, they found something totally different, a real biological plot twist.
Okay, I'm intrigued. What happened?
When they managed to remove both the K79 and K36 marks at the same time, taking out that whole epigenetic duo, they saw the complete opposite of silencing.
The opposite, so.
The genes went into overdrive. Cheng Zhang actually said, and I'm quoting here, "To our surprise, when we removed the markers, we found that the genes were overactivated."
Wow. So removing two on signals somehow turned the volume way up.
Precisely. This double trouble scenario, missing both marks, didn't quiet things down. It caused genes to hyperactivate, leading to a complete breakdown in normal cell development. It's like all the off switches on the instruction manual suddenly got jammed in the on position. Chaos.
Okay, so how did they actually figure this out? What was the experiment like? The methodology sounds like it must have been quite advanced.
It really was clever. They started with what are called model organism stem cells. These are great because they're like blank slates, right? Cells that haven't specialized yet, capable of becoming almost any cell type. This lets you study those very early decisions cells make.
Makes sense. Start at the beginning.
Then, to precisely mess with these genetic volume knobs, they used CRISPR-Cas9. You've probably heard of it. It's this powerful gene editing tool.
Yeah, molecular scissors, basically.
Kind of like that, yeah. It allowed them to very precisely snip out the genes that produce those enzymes we mentioned, DOT1L and CTD2, the ones responsible for adding the K79 and K36 marks.
And they did this step by step.
Systematically. First, they removed one enzyme, saw the effect. Then they removed the other enzyme, saw that effect. And then the crucial step, they removed both together to see that combined synergistic impact.
Like testing stereo knobs one by one and then seeing what happens when you remove both volume controls entirely.
That's a great analogy. And when they looked closely at the outcomes, what did they observe? Well, first, they measured gene activity across the board using RNA sequencing. That gives you a snapshot of which genes are on, off, or in this case, way too active.
A genetic activity map.
Exactly. Then they looked at what happened when these stem cells tried to differentiate, especially into neurons, nerve cells, and the result was pretty stark.
What did they find?
When both marks were missing, the cells just couldn't become healthy nerve cells. The ability was completely blocked. That developmental pathway was essentially shut down hard.
So the instructions were just scrambled.
Utterly. And they didn't stop there. They dug deeper into the mechanics of why this hyperactivation was happening. They looked at something called chromatin accessibility, basically, how tightly or loosely the DNA was packed. If it's looser, it's generally easier to read.
Okay.
They also looked at the activity of RNA polymerase II. That's the actual machine in the cell that reads the genes and makes the RNA messages.
The gene reading machine. Got it.
And what they found was increased transcription elongation.
Transcription elongation. Sounds technical. Can you break that down a bit?
Yeah, sure. Imagine that RNA polymerase II, our gene reading machine, is like a train running along the DNA track, reading the instructions as it goes.
Okay, like a high-speed train.
Right. And when both the K79 and K36 marks were gone, this train wasn't just reading normally. It was racing. It was speeding down the track, making way too many copies of the genes, much further down the track than it should have.
So it lost its speed limit and its designated stops.
Perfectly put. It was moving too fast, going too far, just churning out excessive gene copies, uncontrolled activation. It really showed how those K79 and K36 volume knobs normally work together to keep things finely tuned, keep that train running on schedule.
That's a remarkable finding. So, okay, this is fascinating basic science, but what does it actually mean for us? Why is this important for, human health and medicine?
Right, the so what question. Well, it has some really profound implications, linking directly to serious diseases.
Like the neurodevelopmental ones you mentioned.
Exactly. The study clearly showed how crucial this K79-K36 duo is for normal development, especially for nerve cells. When that process gets disrupted, like it did in their experiments, well, it gives us vital clues about conditions like neurodevelopmental diseases where brain development or function goes wrong early on.
Understanding the breakdown is the first step to fixing it.
Precisely. And maybe even more immediately exciting is the potential for a completely new treatment angle for certain types of leukemia.
Blood cancer, right.
Mm-hmm.
How does this connect?
The researchers found that in some leukemias, defects in these exact same K79 and K36 marks can stop blood stem cells from maturing properly. Instead of becoming healthy blood cells, they get stuck in this immature, rapidly dividing state. That's a hallmark of cancer.
So the faulty epigenetic marks contribute to the cancer itself.
It seems so. And this leads to a really exciting possibility for treatment. The research pinpointed a specific protein partnership. It's called YAP-TEAD. That becomes hyperactive when both the K79 and K36 marks are missing.
YAP-TEAD. Okay, what does that do?
Think of YAP-TEAD as like a cellular amplifier. When it gets overactive, it takes that already racing gene reading machine we talked about.
A runaway train?
Yeah, and it cranks the volume up even further. It directly contributes to that dangerous gene hyperactivation in these leukemia cells.
Okay, so YAP-TEAD is a problem when these marks are missing. What's the breakthrough?
Here's the really cool part. Drugs that specifically target that YAP-TEAD pathway. They are already in clinical trials for other types of cancer, things like mesothelioma.
Wow, already developed.
Exactly. This is huge because it suggests a path to new leukemia treatments could be way faster than starting from scratch. These drugs weren't really on the radar for leukemia with these specific epigenetic problems before, but now there's a really clear rationale to test them.
So existing drugs might find a new purpose here.
That's the hope. As the lead investigator, Kai-Sian Kao, put it, and this is significant coming from the PI, not only that, but we also think there could be a therapeutic impact. That's a direct line from basic discovery to potential treatment.
That is exciting. But of course, there are next steps, right? It's not ready for the clinic tomorrow.
Absolutely not. It's crucial to emphasize that. This hypothesis, while really promising, needs rigorous testing. First, in preclinical models, more complex systems that better mimic the human disease before anyone considers human trials. That's a critical bridge to cross.
But having drugs already in the pipeline for other things definitely gives it a head start.
A significant head start, yes. It shortens that long drug development timeline considerably.
Now, this research also seemed to touch on some deeper complexities, some nuances, and maybe future questions.
Oh, definitely. Science always opens more doors, right? For instance, they noticed the subtle crosstalk between the K79 and K36 marks.
Crosstalk, what does that mean?
It means the level of one mark could slightly influence a level of the other. Like, if they got rid of the K36 mark, the level of the K79 mark went up just a little bit, and vice versa.
So they're not totally independent operators. They kind of sense each other.
It suggests a more complex, interconnected system than we thought. Like, there are these subtle feedback loops controlling the whole epigenetic landscape. It's fascinating and needs more digging.
And what about context? Does this apply everywhere?
That's another big question. This strong effect removing both marks leads to gene overdrive was seen very clearly in stem cells. These are highly dynamic cells, constantly making decisions. How does this translate to cells that are already differentiated, cells that have their settled identity, like a liver cell or a skin cell?
Do they rely on the same braking mechanism?
We don't know yet. Do these marks play the same critical volume dampening role there? Or is it different in different cell types? That needs exploring.
And you mentioned a kind of chicken and egg question earlier.
Right. Could the changes they saw, like the DNA becoming more accessible or that YAP-TEAD protein getting involved, could those actually be a consequence of the genes going wild initially rather than just the cause?
So the hyperactive genes themselves might trigger things that make them even more hyperactive. A vicious cycle.
Exactly, a self-reinforcing feedback loop. Untangling that true cause and effect sequence is really important because it could reveal even more precise points for potential intervention.
Makes sense if you target the trigger versus the consequence.
And then there's the whole world of the reader proteins, the molecules that actually recognize these K79 and K36 marks that read the sticky notes. We're still learning who all the readers are and how they interact with everything else controlling the genes. This study really just opens up so many new questions about how our genetic instruction manual gets interpreted.
So much uncharted territory still.
Definitely. Every discovery like this just peels back another layer of incredible biological complexity.
Okay, so let's try and distill this down. What's the big picture takeaway from this deep dive?
Well, we unpacked how these two tiny epigenetic marks, the K79 and K36 ones, work together in a really surprising way. They act as crucial brakes or volume dampeners on gene activity, especially during development.
And counterintuitively, when both are missing, things don't just quiet down.
Right. The genes go into dangerous overdrive. This can block normal development, potentially contributing to neurodevelopmental issues. And it looks like it plays a role in certain leukemias by messing up blood cell maturation.
And the potential silver lining is that understanding this mechanism points towards a new therapeutic strategy for those leukemias.
Yeah.
Possibly using drugs that already exist.
Exactly. It really showcases the power of fundamental science. You start by asking basic questions about how cells work, and you end up potentially finding new ways to treat disease. It shows this incredible elegance, but also the profound complexity within our cells. A tiny change, 2 missing sticky notes can have huge effects.
It really is amazing, that idea that removing something actually amplifies the signal. It really sticks with you. What stands out to you listening to this about how these intricate programs work or how their failure leads to disease?
It's a lot to ponder, isn't it? Maybe as you go about your day, just take a second to consider that silent, incredible symphony happening inside every single one of your cells, billions of them, constantly reading and interpreting these instructions.
All orchestrated by these tiny, powerful epigenetic signals we're just beginning to fully understand.
And the more we learn about them, the closer we get to figuring out new ways to keep that symphony playing perfectly in tune.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.